We are now in chapter 2, where we are going to study some fundamental concepts of proteins. And this, again, we're still doing some basic chemistry here just to give you a background of everything. But we're going to try to make things a little bit tailored towards cosmetic science. But essentially, proteins are the building blocks of our cells and tissues. And proteins are made of individual amino acid residues that are basically linked together to form a long chain molecule that adopts this complex three-dimensional structure. So these amino acids, there are 20 common amino acids that are known, and they're referred to as alpha amino acids. Proteins themselves are usually classified as globular or structural, and we'll talk about both of those in this lesson. Globular proteins, the one that you're looking at on this first slide here, this is a globular protein. This is some type of, I think, protease. It's an enzyme type protein that breaks down other proteins actually. But globular proteins are water-soluble aqueous proteins. They have a three-dimensional sort of spherical shape like this one does. And in many cases, when we talk about these types of proteins, we're talking about enzymes. And enzymes are a type of protein, and they actually catalyze important processes in the cell. We also have proteins like hemoglobin, which you've probably heard of before, and it carries out a very important process in the body. And hemoglobin actually transports oxygen from the lungs to the muscle tissue. And then there's another protein known as myoglobin, which binds the oxygen in the muscle tissue and actually stores it there until it's needed for energy. But then, so those are all examples of globular proteins. If we talk about structural proteins, these actually provide the scaffolding for the entire human organism. So what do I mean by that? So structural proteins basically are the proteins that tissues are made out of, organs are made out of, cellular organelles are made out of. One great example would be collagen. Basically, collagen accounts for most of the structural composition of tendons, cartilage, cornea in the eye, bones, and also skin. We'll see in skin that the collagen actually is a chief component of the dermis of the skin. So going back to the 20 amino acids, that's what we'll start with. And then we'll go in and talk about amino acids as buffers. So we briefly went over the titration curve. I believe it was for acetic acid during the first chapter. We'll briefly mention that again, and then we'll show titration curves for some of the amino acids. We'll also discuss how peptides are formed. So we'll basically show you how two amino acids come together to form a peptide. And then we'll briefly mention some small peptides in skin, and then look at large peptides. And large peptides are actually proteins. Then we'll look at actual protein structure. So we'll look at the all different types of protein structure. Actually, we have it outlined here: primary, secondary, tertiary, and quaternary structure. These are very important, and they sort of allow us to understand how a protein has a complex three-dimensional structure. We'll also briefly talk about disulfide bonds and how these stabilize protein structures. And you'll see disulfide bonds are extremely important in keratin tissues, especially. Mainly, you think of hair. Hair is really rich in disulfide bonds. Fingernails and the nail system is also rich in disulfide bonds. So mostly the hard keratins are rich in disulfide bonds. The soft keratins, such as skin, do not have as many disulfide bonds. We'll talk a little bit about vitamin C deficiency, and that's just because we talk about collagen, and collagen is important for structure, basically the architecture behind, as we mentioned, many of the structures in the body. So we'll talk what happens when you have vitamin C deficiency. Then we'll just compare and contrast globular and fibrous proteins, and then briefly mention some things about the determination of three-dimensional structure of proteins. So first, we'll start off with amino acid structure and just, what are amino acids? And this is a figure that was taken out of page 460 in your book. And what we essentially see here is that, these are referred to as alpha amino acids. And the reason for that is because if you look at all of these amino acids, you'll see that they all have something in common. They have a carboxylate group here. You can see I'm circling it with the mouse. And they have an amino group down here. And in between, they actually have a carbon that's present. And this carbon is called an alpha carbon. And that alpha carbon is where you get the name alpha amino acid. So if you look at glycine, this is the simplest amino acid. It only has CH2 coming off of the alpha carbon. But usually this is where what's known as the R group is attached to the amino acid. So if you look at alanine, if glycine is the simplest amino acid, alanine is the second most simplest, or it's a little bit more complicated or complex. Essentially, alanine has, the alpha carbon is here again, and it just has a methyl group coming off of it. If we look at valine, we can see that in addition to a methyl group, we essentially have a methylene, or a CH group here, which is attached to two methylene groups here. So in these particular aliphatic amino acids here, you can see you start with glycine, go to alanine, valine, leucine. The structures become a little bit more complex and actually a little bit more aliphatic per se, which means they, when you talk about aliphatic chains and things like that, you're talking about long alkyl type chains, and they actually they're sort of things that interact in hydrophobic environments. So again, you have the carboxylate present and the amino group present in all of the amino acids, and then you have this alpha carbon in between them, as I'm pointing right now, and from that alpha carbon you have an R group that comes off of that, and each R group is distinct for all of the amino acids. So the alpha carbon is actually what's known as the chiral center for these amino acids, and that's the reason we say that is actually these amino acids have two possible stereoisomers. They can either be in the D or the L state. These isomers are essentially non-superimposable mirror images of each other. Okay, and they are also referred to as enantiomers. Enantiomers, ENANTIOMERS. So these stereoisomers, this DL system, this was actually designed by Emil, EMIL, Fischer, FISCHER. And you probably remember studying Fisher projections when you studied carbohydrate chemistry in your organic chemistry class in undergraduate. So this DL system does not necessarily refer to levorotatory or dextrorotatory, which are normally used to designate optical behavior. So again, they do not refer to levorotatory or dextrorotatory. The reason we point this out is because in biological systems, when you have isomers or enantiomers present, it's extremely critical because it depends on the stereoisomeric form whether or not a particular, in this case, amino acid or any type of biological molecule is active, and if it can actually carry out its function. So if you have an L versus a D isomer, one or the other might not be able to carry out its function because it's not the proper conformation or the proper stereoisomer that has to function properly in nature. Okay. In proteins, all of the amino acids are L stereoisomers. Okay? Again, in proteins, all of the amino acids are L stereoisomers. Now, in contrast to that, when a molecule is synthesized in the laboratory, you end up with a racemic mixture of both the D and L stereoisomers. So again, that's, if you try to use this synthetic mixture, not all of it's going to work. Only the L stereoisomers are going to work if you try to use it in a biological system. So back to the division and sort of understanding here of the amino acids. We have these amino acids can essentially be, they have distinct structure, size, electric charge, and either hydrophilic or hydrophobic characteristics. Okay, and again, with the exception of glycine, which has two protons or two hydrogen atoms coming off of the alpha carbon here, all of the other amino acids have 4 different groups coming off of the alpha carbon, and that again is why with the exception of glycine, all of the other amino acids have a chiral center at this alpha carbon position. Okay, so we start off again. We already mentioned this first group up here in the top. These are the aliphatic amino acids, and you can sort of see why they have these sort of aliphatic type groups attached in the R position. You can also have amino acids that have polar groups. So you have polar amino acids with uncharged R groups. So this group, you can see that you have, for example, hydroxyl group, mostly the hydroxyl group. You can also see that you'll have a sulfhydryl group. Sulfhydryl is SULFHYDRYL, sulfhydryl. You also have, both amino and a ketone type function here. So this is actually an amide that you can see here, both in asparagine and glutamine have this type of structure at the end of the R group. Then we have amino acids that can either have a positive or negative charge, and that we're talking about the R groups, not at the moment, the negative and positive charge that are shown up here with the carboxylate group and the amino group. These can change, if the amino acid is by itself in solution. But if it's part of a peptide chain, as we'll see, that's not going to be the case, and you'll see why in a few minutes. But when we're just talking about the R groups, we can have negative or positively charged R groups. Here we have the acidic amino acids that are shown in the middle here. You can see these have negative groups. The carboxylate groups at the end of the R groups are negative. And again, this depends on the pH of the medium that you're working in. We also have the basic amino acids. These can have a positive charge. And again, you can see that we have lysine, arginine, histidine. These all have these amine functionalities at the end. And again, what's shown on this slide is at neutral pH. So at neutral pH, this is the charge that you would expect to see here. Same with aspartic and glutamic acid. This is at neutral pH. That's what you would see. And for all of the individual amino acids, when we write it down as neutral pH, we have a negative charge here, positive charge here. Okay. Another group of amino acids are the aromatic amino acids, and here we have the three important ones. Phenylalanine is the most simple of them. It essentially has a methyl group that's attached to this phenyl group or this aromatic ring. The next one in complexity is tyrosine, essentially the same type of molecule with an addition of a hydroxyl group here. And then we have tryptophan, which is unique in its own way, not only structurally, as you can see here, it has quite a complex structure. It has amine functionality within the structure. But when you look at these, all three of these molecules absorb UVB radiation, and they all fluoresce. However, tryptophan has the highest molar absorption coefficient, the highest molar absorption coefficient, and it actually yields greater fluorescence than phenylalanine or tyrosine. So it also absorbs more, and it fluoresces more than the other two. And again, this is because it has the higher molar absorption coefficient. The side chains for these three molecules basically renders them hydrophobic, and they essentially associate with other hydrophobic moieties in proteins through hydrophobic interactions. Okay, so just to sort of give you a better illustration of that, this is a UV absorption profile, or actually it's just, you can call it a UV vis absorption profile, but it's only showing light in the UV range for both tryptophan and tyrosine. So this is actually going from UVC light, which is down to 230. When we get up to 280, that's the end of UVC. Then you start to get into the UVB region. Actually 290, I believe. 290 to 320 is the UVB. And then 320 to 400 is UVA, and that isn't shown on this since these two molecules don't really absorb in that region. But what you can see here is that right around 280, even a little bit to the right of 280, you can see that peak maxima occur for both tyrosine and tryptophan. And what you'll notice is that tryptophan has a much greater absorption, and again, this is due to its higher molar absorption coefficient. So just to give you a little bit of some background on what you could do with understanding absorption and fluorescence of tryptophan, this is actually showing you a fluorescence emission spectrum of hair. And So fluorescence is, a little bit different from absorption. Absorption, we're just going to talk about tryptophan right now. Tryptophan absorbs light with a maximum of 285 to 290 nanometers. So it absorbs light in that region. So so essentially what you're doing is you're shining light on tryptophan and it's absorbing it. It's basically capturing that light with the aromatic structure that it has. Now, what happens to things when you shine light on them? Well, they can either absorb the light and they can dissipate it as heat, as thermal energy, so as heat. Or what a lot of things do is not a lot of things, but some things actually fluoresce. And tryptophan is one of those things. So it absorbs light around 285 to 290 nanometers, and then it fluoresces light at wavelengths that are a little bit longer than that. So if you look here at this curve, it's showing you the maximum of the fluorescence of tryptophan, which is, I don't know, this is probably around 330 here or something like that. This is tryptophan fluorescence, and then you can see that's where the maximum is, and then it sort of starts to die off here as you get past 410 into this region. So this is actually, this spectrum that we're looking at, this is actually a spectrum, a fluorescence emission spectrum for hair. And this is virgin hair that's in good shape. And in this hair, what we would expect is that the proteins are in good shape. If we say the hair is in good shape, that means the proteins are in good shape. But if we damage the hair, we damage the proteins. And this bottom curve essentially is showing you an emission spectrum of tryptophan in hair for hair that was exposed to UV light for an extended period of time. And what you essentially see is that when you take hair and you expose it to UV light, like, I don't know, for 72 hours or something like that, if you come back and read the fluorescence of tryptophan, you'll see that it drops from its initial state. So it goes from up here to down to here. And why? Because UV light essentially damages tryptophan. That's pretty well known. And when it damages tryptophan, it essentially damages the rest of the protein structure as well. So we're using tryptophan as a probe to know, what is the health state of the protein in the hair. And you can see here, it's not very good. So again, this is for tryptophan for virgin hair. This one down here is for tryptophan that was exposed for an extended period to UV light, and you can see that the emission of tryptophan is much less. So there's a lot of protein damage in this bottom sample here. What you'll notice is that these two spectrum cross over each other, and that's because there's a second peak out here, and this second peak is actually due to the presence of compounds known as kynurenines. It's KYNURENINE, kynurenine. Let me spell that again. It's KYNURENINE, kynurenine. If you need verification for that, send me an email, but I think that should do it for you. But it's okay, so the kynurenine is out here. So kynurenine is actually a degradation product of tryptophan. There are two different degradation products of tryptophan. One is kynurenine, the other is melatonin. Melatonin is a molecule that makes you sleep. When everyone talks about going to Thanksgiving dinner, eating turkey, and then later becoming tired. The reason they become tired is because turkey has high levels of tryptophan in it, and when you eat or ingest tryptophan, it is metabolized to melatonin, and the melatonin is essentially what makes you sleepy. Melatonin, again, is one degradation or metabolic product of tryptophan. The other degradation product or metabolic product is kynurenine. So when we say degradation product, we mean, okay, if we take tryptophan and we expose it to UV light for an extended period of time, we're going to form kynurenine. So tryptophan is going to be damaged and degraded, and we're going to have the formation of kynurenine. And that's what you see on this plot. Essentially, in the first one, you have low levels of kynurenines in the virgin, unexposed hair, unexposed to UV light. But then, the one that's exposed to UV light, when the tryptophan drops over here, we actually see an increase in the kynurenines. And that increase, again, is because tryptophan is degraded to the kynurenines. Okay, I hope that that's somewhat clear. I know it's a little bit complex the first time seeing it. Please understand this part well, and if you have any questions on this, please do get back to me. Okay, so in addition to the 20 common amino acids that we already looked at, just talking about, we're going to talk about collagen a little bit, and there are certain amino acids in collagen that are important. Collagen is made up of three principal amino acids: glycine, proline, and hydroxyproline. Okay, and here we have the structure of hydroxyproline. You already saw the structure of proline. Essentially, hydroxyproline just has a hydroxyl group that's attached to this 5-membered ring here. So this is one of the amino acids that's present in collagen. There are three principal ones: glycine, proline, and hydroxyproline. Now, collagen is a fibrous protein, and so is elastin. Elastin is another important biomolecule or structural protein that is present in the dermis of the skin. And we can see here we have this molecule known as desmosine. So this is another type of amino acid. Again, hydroxyproline is also, so these aren't the 20 normal amino acids. And what you can see here is this essentially is a crosslinker between elastin chains. So essentially these 4 points where you can see the carboxylate groups here, these actually get attached into different chains of elastin, so that you build a network of all the different chains of elastin, and it overall has these physical properties that give it a very elastic nature. So just like a rubber band, you stretch it and then it snaps back into its original position. Okay. This is, again, a type of crosslink between elastin. Another type of crosslink that we are going to be interested in learning this semester is the disulfide bond, and that's formed by 2 cysteine residues. We'll talk about that. And when you have 2 cysteine residues come together, they result in the formation of cysteine. Let's just go back for 1 second to the slide here, you can see this amino acid right here that you're looking at. This is called cysteine. It's spelled with E-I-N-E. And the other one that I'm referring to, the actual crosslinks, are when crosslinks are formed between two of these molecules. Essentially, you'll have two sulfur groups coming together. It's called cysteine, and it's spelled C-Y-S-T-I-N-E. C-Y-S-T-I-N-E. So, in all of the amino acids that we've sort of talked about so far, the structure is shown when the amino acid is in a neutral aqueous solution. So this is the Zwitter ion form, which is amphoteric form, meaning that you have both negative and positive charges in the same molecule. Again, the carboxyl terminus is deprotonated. It has a negative charge in this form, and the amino terminus is protonated and actually has a positive charge. So, as I mentioned, this is all pH dependent. So if we were at acidic conditions, therefore low pH, the carboxyl terminus would actually be protonated, so it would be neutral, and then we would have an overall positive charge on the amino acid due to the proton on the amino terminus. Okay. So in contrast, if we are working in a basic medium at high pH, the carboxyl would be deprotonated, and so would the amino termini, the amino groups. Okay, and in that case, we would have an overall negative molecular charge. So in chapter 1, we introduced the concept of weak acids, weak bases, buffers, and pKa. And I think it was mentioned that amino acids that are present in proteins, these contain weak acids and bases as their R group. And these R groups play a very important role as physiological buffers. Okay? Now, as I mentioned before, when we talk about proteins or peptides, the pKa, we mentioned that during the last class, the pKa of the carboxyl and amino termini, so essentially the carboxyl portion and the amino portion, the pKa value isn't as important because if you're talking about peptides or proteins, you have the amino acids linked together, so there's no charge, at the end of each amino acid. Just at the very end, the carboxyl terminus or the amino terminus of the entire polypeptide chain. But when we talk about R groups, the pKa is more important since, this is how the molecule is going to exist in the protein, and that R group is in many cases ionizable, meaning that it can change charge depending on the pH. Now, not all of them can, but the ones that contain the carboxylate groups or the amino groups can change charge depending on the pH. One thing to sort of bear in mind is that pKa essentially provides us with an indication of the tendency of an amino acid to give up a proton. So low pKa means the proton is not held strongly, while high pKa means that the proton is held with a greater affinity. Okay. Another sort of definition, and we'll see this in some of the things we're talking about in just a second, the isoelectric point in a protein is the point, or it's the pH of the molecule when the sum of the positive and negative charges is 0. So let's look at some titration curves just to help us understand some of this a little bit better. So again, the one that's shown on the screen right now is the titration curve of acetic acid. As you can see, we discussed this during the last lecture, and you can see that at low pH, or actually at low quantities of added base, you have acetic acid sort of in its protonated form, so the COOH group is there. As you add hydroxyl groups to it, the pH increases, and then you sort of enter this blue zone, and this blue zone is known as the buffering region. That's between pH 3.76 and pH 5.76. And of course, this depends on the molecule, the characteristics of the molecule. And when we get to this sort of midpoint of the titration, which is shown here, at that point, we essentially have the same concentration of protonated carboxyl group as we do deprotonated carboxyl group. So you can see here equivalent concentrations of those two. As you proceed adding hydroxyl equivalents, or actually adding base, we should say, you can see that you end up in this sort of deprotonated form eventually. So that was for pretty basic molecule, but now let's look at the amino acids, because I mentioned that amino acids essentially serve as buffers. You could sort of say in the cytoplasm of the cell, they help maintain the pH in certain regions. So glycine, just to give you some characteristics of that, if you do a titration curve of glycine, you'll see as you start to add base to that, you don't really get a large shift, and you actually go through this first stage where you have one buffer region, and in this region it has pK1 equals 2.34. So depending on how many ionizable groups there are on the amino acid, that's how many pK values you'll have. So for glycine, you essentially, it has an R group of H, or hydrogen, so you just have the possibility of ionization at the carboxylate group down here or the amino group up here. So when you're at low pH, this is the carboxylate group is already protonated, so is the amino group, so you have an overall charge of 1. As you add base in this area, you can see that the pH doesn't change drastically. So again, this blue section is all a buffer region. But then you get to a certain point where you add a little bit of base, and then the pH shoots up very quickly. So you can see the corresponding structures. In this first zone over here on the left, we have protonated amino group and protonated carboxyl group. But then when you go to this middle region here, in the middle region here, we have a protonated amino group and a deprotonated carboxyl group. So the first pK value corresponds to the removal of the proton from this carboxylate group here. Okay. So essentially this low pKa value is 2.34, and what you'll see is that as you go and you continue to add base to this system, you rapidly rise, and then you enter into the second buffering region here. And it's during the second buffer region that we actually see the conversion begins to take place from this middle molecule here, so plus and minus in the neutral state, and conversion to one in which you have deprotonated NH2, and this carboxylate group is still deprotonated. So you essentially go through this whole process of titration. You remove a proton first from this carboxylic acid group, and then secondly from the amino group to end up with NH2 and COO minus. So the second pKa value is 9.6. So you can see that if you go through a titration with glycine, the first pK of the COOH group, the proton here is easier to remove than removing the proton from the amino group. Okay. We also show the isoelectric point here is 5.97, and that's where we have an equivalent concentration of both the positive and negative charges in the molecule. So let's look at something a little bit more complicated, and this is glutamate. It's also called glutamic acid, and essentially, so you can see we have typical amino group and carboxylic acid group here. Then we have the alpha carbon that's shown here, and then coming off of the alpha carbon we have the R group, so we have CH2, CH2, COOH. So at low pH, this is the predominant form. We have a protonated amine group and a carboxylic acid group that also has its proton. As we begin to add base, what happens? Well, we start the conversion from this first structure to the second structure, and just like before, the carboxylic acid group more easily gives up its proton and becomes deprotonated. So you can see that that's what's happening in this first area, and there's the first pKa value of 2.19 corresponding to that process. Then we go from the next step, we continue to add base, and eventually what happens is that this one already is deprotonated up here. Then the R group carboxylic acid becomes deprotonated, and that's what occurs in this middle region here. So again, this is sort of like a buffering region. It's not highlighted very well in this figure, but essentially that's what's going on here. So we have a pKa value of 4.25, and we end up with two deprotonated carboxylate groups. As we increase the amount of base in the titration, you can see it sort of moves along here steadily, and then at some point it's a very rapid increase in pH, and then you enter this third zone, and then in this third zone is where you're going to have the transformation, and it's also the buffering region, and it's the third buffering region. But you'll have the transformation from this protonated amine group to a deprotonated amino group. So you can see that now we're in the fully deprotonated form, shown here on the right, and the pKa associated with removing the proton from this nitrogen is 9.67. Okay. Again, equally complex. That was for an acid, or an amino acid that had an acid type R group. Here we have an amino acid with an amine type R group. You can see here, this is again showing you have the carboxylic acid portion, you have the amino group, you have the alpha carbon here, and then you have the R group, which is the methylene group here, followed by this 5-membered ring type structure with this amino functionality in it as well. So in this case, as you go from, protonated amino group, um, protonated carboxylic acid group, you start to add base. You can see this first sort of pKa transition is shown here at 1.82, and again, this is like a buffer region, even though there's no blue box here. The first thing that happens is you remove the proton from the carboxylic acid portion, and then as you continue to add base, you sort of have an area or region where you add base and you get pretty big changes in pH, and then you sort of enter this second buffering region that's shown here, and that's the transition from the second structure to the third structure. And in this transition, we're going to actually have this nitrogen give up a proton, and the pKa associated with that is 6.0 shown here. We're going to add base, continue the titration, and then we'll enter into this third region. And again, this is like a buffering region right in here. And in this third region, we're going to have the amino group here give up a protein. So going from NH3 plus to NH2, and then that's going to have the pKa of 9.17. So you can see that, these are looking at individual amino acids. These are the pKa's associated with removal of each of the protons or hydrogen atoms. And one thing to keep in mind is that these are for individual amino acids. These aren't for peptides. So again, in peptides, the story is going to be a lot different, because you're not going to have any charges on, you're going to only have one charge at the very end of the polypeptide chain on one side of it, and then you're going to have another charge on the other end. So you're not going to have each amino acid with charges at the carboxylate group here and the amino group here. So the R group is what's really going to dictate what happens with that. Okay, so now that brings us to talk about the formation of a peptide bond between two amino acids. And again, here you can see we have two generic amino acids that are shown here. So we have the carboxylate group, the amino group, and then the R group. These are present in both of these. We need water to form a peptide bond. So, essentially this reaction takes place between these two, and you end up with the peptide bond between this carboxylate oxygen, so the very end oxygen here, and then between the amino groups. So you end up with, removal of the proton from both, actually. You don't have protons on either one of these, so you have NH2 and O. So, this is a fairly unique bond, and, one of the reasons, it's a very rigid bond, actually, and there isn't really a lot of free rotation about that bond, and it actually makes this whole region, starting from nitrogen, going through the peptide bond, through this oxygen, up to this carbonyl oxygen. It makes this all very rigid, and these are all sort of in the same plane. So there's actually some electron resonance that occurs as a result of this, and electrons are shared in between these three molecules rather easily. Okay, so even though that the rotation is restricted around the peptide bond, free rotation does occur between the carbonyl carbon and the alpha carbon. So the carbonyl carbon is here, the alpha carbon is here. There's free rotation about this bond. The same occurs over here between the alpha carbon here and the carbonyl carbon here. This is also true between the amide nitrogen and the alpha carbon. So the amide nitrogen is here, the carbonyl here, so there's free rotation about this bond, and we also see that there is free rotation between the alpha carbon and the amide nitrogen, which is shown here. One of the things that lets us know the two-dimensional, the secondary structure of proteins, which we'll sort of talk about that a little bit later, but if we look at like dihedral angles of this, if you remember dihedral angles from organic chemistry, you could do like Fisher projections and things like that. And from the dihedral angles, one is called phi, phi, that's the Greek letter phi, and the other is psi, psi. And essentially, the dihedral angles that result from the carbonyl carbon and the alpha carbon and the amide nitrogen and the alpha carbon, these essentially determine the peptide secondary structure. So again, the dihedral angles, the first one is called phi, phi, and the phi angle is basically comes from the dihedral from the carbonyl carbon and the alpha carbon, and the psi, psi angle, or the dihedral, comes from the amide nitrogen and the alpha carbon. And essentially, depending on what these dihedral angles are, that determines protein secondary structure, and things like whether or not you have alpha helix, beta sheet, or random coil structure, and we'll talk about those a little bit more in depth in just a few minutes. So let's just talk a little bit about small peptides in skin. And there are many large proteins found in skin, but there are also a number of small peptides, and a lot of these serve as antimicrobial agents, and they're located close to the skin surface, and they actually play an important role in the skin's natural defense system, and they constitute the flora, Flora, of the skin. So these are like, when we say flora, we usually think of bacteria, but they can also mean other protective agents such as these antimicrobial peptides. And in addition to this, that's one role for these small peptides in skin, but there's a second role, and that's a role as hormones. And essentially, hormones are synthesized in one part of the body, and then they travel to distant locations where they essentially act as signaling molecules. So, hormones travel from one spot to another, and they sort of stimulate some type of response in the body. So, just with particular reference to skin, we're going to just make note of this tripeptide here. It's called glycyl histidyl lysine, and this was more meant to give you an example of how amino acids are joined together to form a peptide. This is a small peptide. Again, it only has 3 amino acids, and this particular peptide, it actually circulates in human plasma, so in the blood, and it plays a very important role in wound healing and tissue repair. So, it works by somehow complexing copper 2 ions, and it's actually known to stimulate or actually upregulate collagen production in fibroblast cells. Okay. So just getting to the formation of peptides and how peptides are named, essentially peptides are named from left to right. So if you look at this peptide, we have Glycyl, so we have a glycine group on the left. Histidyl, we have a histidine group in the middle, so you can see that histidine is characterized by this 5-membered ring that contains the nitrogen compounds or components. And then we have lysine, which is the last or the third amino acid, and lysine basically has 1, 2, 3, 4 methylene groups, and then it has this amino group at the end. So, we start from left to right for the naming, and then the way that it's drawn is that the amino terminus, or the amino terminal residue, it's also called N-terminus, it's on the left side, so you can see here that the N-terminus is on the left side, okay, and then the COO terminus, or actually it's called C-terminus, or you could also call it the carboxyl terminal residue. So C-terminus is on the right side. So N-terminus on the left, C-terminus on the right. Okay? So that's how it's drawn, and then you name it just by going from left to right, picking out, using the names of the amino acids. But you truncate them, so instead of glycine, you have GLYC, and then you add YL to the end of them for all of them except for the last one. The last one, lysine, you use the actual name of the amino acid. So I think, so in general, there are other functions and things for small peptides, but I just wanted to give you an example of how one is put together. Let's talk about large peptides a little bit. Large peptides, we can just say proteins because that's what they are, proteins. Large peptides or proteins are classified as being globular or fibrous, as I already mentioned. Globular proteins, we already said this before, oftentimes serve as enzymes. Enzymes are like catalysts for biochemical reactions. Globular proteins also serve as transport proteins. So an example of that we talked about a little bit was hemoglobin. We also have hormones. That's another function for globular proteins. And actually, the one that we show here, glycyl histidyl lysine, this is actually a hormone that's shown on this slide. And then globular proteins are also very important in the immune system function. And they actually, in that case, are antibodies. Now, so those are all globular proteins or examples of globular proteins. Let's talk structural proteins. So structural proteins consist of things like collagen and elastin, which are found in the skin, specifically in the dermis of the skin. And then we also have things like keratins, which are found in the hair fiber and the nail structure. In addition to this, we also have things in the body like muscle fibers, and these are basically composed of protein fibers. The two principal components of that are actually actin and myosin. Actin, ACTIN, and myosin, MYOSIN. And in addition to that, in addition to those structural proteins and globular proteins, we also find lipoproteins in the skin. You spell that LIPOPROTEINS. So lipoproteins. Lipoproteins are essentially when you have lipids and protein are sort of joined together as one molecule, and they form a combination, which is a lipoprotein. We also have glycoproteins, and glycoproteins, as you can imagine, glyco represents sort of carbohydrates are present, and protein means there's protein present, so you have a combination of carbohydrate and protein to form glycoproteins. And then we also have metalloproteins. Metalloproteins, metalloproteins, and that's when we have a metal atom that's present in the protein. And just for clarification, glycoproteins is spelled GYLCOPROTEINS. Okay, all of these proteins carry out vital biological functions that wouldn't be possible without, the presence of either component. So they play a really important role in human physiology. Now, again, as we said, there are many small peptides in the body, but there are also much larger ones. An example of this would be cytochrome C. It's spelled CYTOCHROMEC. Cytochrome C has 104 residues. 104 residues, so that's amino acid residues. It has a molecular weight of 12,400 kilodaltons, and it consists of one polypeptide chain. As another example of a larger protein, it's actually larger than cytochrome C, we have hemoglobin, which has about 574 residues. 574 residues, those are amino acid residues. It has a molecular weight of 64,500 kilodaltons, and it consists of 4 polypeptide chains. Cytochrome C actually plays an important role in mitochondrial electron transport chain, or in the mitochondrial electron transport chain. That's something that we talked a little bit about before. And it actually leads to the production of ATP, adenosine triphosphate. Okay? And again, that occurs in the mitochondrion, and that's involvement of cytochrome C. Hemoglobin, I didn't spell it, is HEMOGLOBIN. This is essentially, as I mentioned before, responsible for the transfer of oxygen from our lungs to tissues via the bloodstream. Essentially, the protein itself has a prosthetic group that's bound to it, and the prosthetic group is actually what's known as the heme group, or it's like a porphyrin type ring, and it essentially binds the O2. And in that porphyrin ring, or that heme ring, we have an iron atom, and the iron atom is actually what actually physically binds the oxygen. And the reddish brown color that you see of blood is actually due to the presence of hemoglobin in the blood. When you have, and that's in its oxygenated form, when you have blood that's sort of deoxygenated, it changes color, and it actually has a more bluish tinge to it. That's how you can sort of see someone who doesn't have such deoxygenated blood. They'll sort of have this pale bluish color to them, and you can see that through the skin. So you can see, even just with the skin, optically you can see the effects of hemoglobin. The color sort of influence that it has on the skin. So in general, there's kind of like a hierarchical organization of proteins, and you can usually divide that into 4 groups, as I mentioned before. We have primary structure, and primary structure is essentially the sequence of amino acid residues of the polypeptide. So that is, if we go from left to right, what is the sequence of amino acids from left to right that are linked together? So that's primary structure. The secondary structure refers to the local 3-dimensional structure of the peptide chain. And examples of secondary structure include alpha helix, beta sheet, and beta turn. Then we have tertiary structure, which essentially is the overall 3-dimensional folding of a single polypeptide chain. Okay, one polypeptide chain. Now we mentioned that cytochrome C had one polypeptide chain, so that's actually one subunit. But then we have something like hemoglobin that has 4 polypeptide chains, and that has 4 subunits. So tertiary structure is just the overall 3-dimensional folding of a single polypeptide chain. And then if we talk about quaternary structure, quaternary structure actually describes the spatial arrangement of subunits relative to one another. So if you have cytochrome C, it's not going to have quaternary structure because it only has one subunit. But if you talk about hemoglobin, it has some type of quaternary structure because it has 4 subunits. Okay. Just a few things to keep in mind is that the amino acid sequence, so the primary structure, actually determines the 3-dimensional structure. And the reason for that is that the sequence of the amino acids, how they interact together, if you have one next to another, how the protein or polypeptide begins to fold, while when we describe it, it's actually 3-dimensional structure, but how you arrive to that point all depends on the primary sequence of the primary structure or the amino acid sequence. So let's briefly talk about disulfide bonds. Disulfide bonds, we mentioned before, we have cysteine, which is the actual amino acid, and then we have cysteine, which is the cross-linking portion. Again, cysteine is spelled Cystine, and cysteine is when you actually have 2 sulfur atoms covalently bond together to form a disulfide bond, which is like a cross-link between different chains in the protein. It could either be a cross-link between one part of a peptide chain and another, or it can actually be between 2 peptide chains. And one of the things that it does, the disulfide linkage, it actually provides extra stability for the protein, and it helps it from denaturing or unfolding. So, when we talk about globular proteins, and specifically intracellular globular proteins, so inside the cell, these typically do not have disulfide bonds are not very common in globular proteins that are in the cell, but they are found in globular proteins that are secreted into intercellular space, such as cell signaling type proteins. And that's sort of, you can imagine why, is that if you have a protein that leaves the cell, it's going to have a much more robust structure in order not to denature or to become damaged by things outside of the cell. Another place where you'll find disulfide bonds is in structural proteins. And the example that we're going to use a lot in this course are keratins. The keratins have a lot of disulfide bonds in them. And just in general, a general statement is that if you look at the environment in a normal cell, it's a rather reductive environment. So, if you have a reductive environment, that's going to sort of favor the presence of cysteine residues rather than cysteine or disulfide bonds. We can also talk a little bit about archaea. We mentioned those at the beginning of chapter 1. Archaea, again, it's spelled A-R-C-H-A-E-A, A-R-C-H-A-E-A, and these contain disulfide bonds. And as you can imagine, when we talked about the archaea, those are organisms that live in very harsh environments. So, they're going to need the extra support of disulfide bonds so that they're the proteins that are present in these very extreme conditions aren't going to denature. Usually disulfide bonds aren't that common in bacteria unless it's something like thermophilic bacteria that are exposed to high temperatures. So in hair, again, we have disulfide bonds that really provide an important function for the incredible structural strength of the keratin fiber. Essentially, the disulfide bonds can undergo oxidation or reduction reactions, and these are not such good things to happen. So, just looking at it here, we have a little illustration that's shown. So this is showing you a disulfide bond between two adjacent polypeptide chains. So this would be one chain over here, this would be one over here. We would have these cysteine residues which would form together. This is actually a cysteine group that's shown here now. But you can see, you have the amino group, the carboxylic acid, or the carbonyl now. So the C and the N portions of the amino acid. Then you have the alpha carbon, and then we have the side chain that's forming this covalent crosslink between the two peptides. So in hair, or you can have this happen in any type of proteins, but we can have oxidation occur. So, just in general in proteins, if you use performic acid, you can oxidize, and essentially you can break the disulfide bond that's here, and you can see you end up with these, oxygens with the negative charges on them. This is something that actually is very typical in when you bleach hair. So you have the formation of the what's known as sulfonic acid groups. And this happens again when you bleach hair. This hair can undergo this oxidation process, and you end up with a lot of this forming in the hair. When you actually reduce hair, and these are two typical agents that are used to reduce hair. One is dithiothrietol, the other is beta-mercaptoethanol. And when you do that, you reduce hair. You can also break the bonds between the covalent bond here, the disulfide bond, and you end up with cysteine residues, as you can see here. So you end up with the sulfhydryl groups that are shown. And this is actually a process that's employed during permanent waving of hair. And actually a better example of that is shown here on this slide, where we have we start with natural hair. You can see it has sort of these disulfide crosslinks at certain points. It also has sulfhydryl groups at certain points. Um, we stress the hair and we distort it on a roller, for example, or some type of curling device, and then we actually at this point, after we have it distorted on the roller, then we treat it with the reducing agent. So that's when we treat it with the dithiothreatol or the beta-mercaptoethanol. And then from that point, you would remove the disulfide bonds. So you can see that the disulfide linkages are broken at this point. But then they're broken and you sort of have the hair in this new arrangement. And then you actually reform disulfide bonds by oxidation. You actually use hydrogen peroxide to oxidize it, and you can see that you form disulfide bonds. And usually when you reform them, they're not in the same place because the fiber is going to be stressed. So you're going to have different sulfurs joining together again, and then you're going to maintain the new structure that you formed the hair into. And in this procedure it doesn't have a high degree of fidelity. And what I mean by that is that, for each disulfide bond that you damage in the first step, when you oxidize it back to reform disulfide bonds, not all of them are going to be reformed, and often they're reformed but with other cysteine groups in the peptide chain. Okay, so that brings us to the section on three-dimensional structure of proteins. And just in general, the amino acid sequence of a polypeptide determines its three-dimensional structure. Then in turn, a protein's function will be determined by its three-dimensional structure. So we know that overall, a protein's conformation is stabilized by non-covalent bonds. And essentially when we say conformation, the conformation describes the three-dimensional spatial arrangement of atoms in a protein. So proteins in general can be in the folded state, that's their usual functional form, or they can be in their unfolded state. The folded state is essentially an organized structure that is stabilized by disulfide bonds and non-covalent bonds. Okay, we already talked about disulfide bonds. Non-covalent bonds refer to hydrophobic interactions, electrostatic interactions, and hydrogen bonds. The unfolded state of a protein, it's basically analogous to a random coil structure, and that's essentially when the molecule has lost its highly organized three-dimensional structure. So random coil means essentially that the molecule has lost its hierarchical three-dimensional structure. Okay, and then back to the non-covalent interactions. The hydrophobic forces, or hydrophobic bonding, these are probably the most influential in determining protein structure. And usually you find hydrophobic constituents, or R groups, for example, in the interior core of a globular protein. So the hydrophobic groups tend to go towards the center, and then actually the hydrophilic groups, or what we know as the amino acids that contain polar substituents, those are actually the polar substituents and polar type amino acids are actually contained or located on the exterior surface of the protein, and that's because then they can actually interact with water, which is the solvent in the cell, and then that leaves the nonpolar components to be hidden in the interior of the protein structure. So when we talk about protein structure, um, we've already talked about the primary sequence of protein structure, and essentially, how we have a sequence of amino acids, and that sequence corresponds to primary structure. But we also have secondary structure, and that secondary structure is the result of the spatial arrangement of the atoms on the backbone of the peptide. So, as we mentioned before, secondary structure is determined by the dihedral angles, and remember there are two of them. We have phi, phi, which describes the angle that's the dihedral angle between the carbonyl carbon and the alpha carbon, and then we have the psi dihedral angle, psi, which is the dihedral angle between the amide nitrogen and the alpha carbon. The most common secondary structures are alpha helix, beta conformation, and beta turn. And again, we already mentioned random coil. Random coil isn't actually, I wouldn't really say it's a type of secondary structure. It just indicates that the polypeptide has no secondary structure. Okay, so now, we're looking at an alpha helix on the slide in front of you right now. Alpha helices in general can be right-handed or left-handed. And the twist of the molecule, it's either when you look along the axis of it. So if you, took the alpha helix that's shown in part A of the figure here, you rotated it on its side and you looked at how the helix was wrapping around the pole. It's an imaginary pole to show you where the axis is. It could either be clockwise or counterclockwise. And typically, in a single turn of an alpha helix, you would find about 3.6 amino acid residues. And the distance, as you can see, that's noted there in a typical turn, is about 5.4 angstroms. The dihedral angles for an alpha helix structure is psi is negative 57 degrees. Phi is negative 57 degrees, and psi is negative 47 degrees. And essentially, with an alpha helical type structure, the interior, like where the pole is located at in this diagram, that's where you have hydrogen bonding occurring. So you have, groups that can participate in hydrogen bonding on the interior of the alpha helical structure. Now, the substituent groups, or the R groups of the amino acids, are actually located on the exterior of the helix. So the hydrogen bonding that occurs actually occurs between the carbonyl oxygen and the hydrogen atom attached to the amide nitrogen. Okay, the hydrogen atom attached to the amide nitrogen. So these are on the backbone of the polypeptide chain. And these are relatively, so that's going to be common to most polypeptides. Now, in general, certain atoms or certain amino acids actually promote alpha helix formation, and there are others that actually prevent it. Alanine is a good example of a molecule that fosters or helps in the formation of alpha helices. But if we look at proline or glycine, on the other hand, these strongly deter alpha helix formation. And one of the reasons why is that, in proline, if you recall back to the structure that we discussed before, the nitrogen atom is part of the ring structure, and it basically impedes free rotation about the alpha carbon nitrogen bond. And so between the alpha carbon and the nitrogen bond, we have restricted free rotation, and that's why proline deters alpha helix formation. And glycine, in general, it's the most simple amino acid. It just has a substituent group of hydrogen. It tends to form highly coiled structures. So in this diagram that we show here, it's just to show you how an alpha helix is typically formed. If this is a ball and stick model that's shown here, so it just shows the atoms that are attached. The balls are the atoms, the sticks are the bonds. If we rotate this towards us, we can sort of look down the axis, and it appears that there's a hole in the middle of the axis, but this actually, if you plotted this, or if you illustrated it with a van der Waals or space fill model like the one shown in C, this would fill up significantly here in the middle. And again, remember, this is where the hydrogen bonding takes place as well. It's in this area along the axis here. So C again is the van der Waals or space fill model. So it gives you a better idea of how much space the atoms are actually occupying in the molecule. And then in D, this just shows you an atom drawing representation, but it's kind of nice because it sort of cleans things up. And this one in particular shows, how if you have amino acid at the number 1 position and then the amino acid at the number 4 position, if you had a positive charge on the substituent group at the 1 position, a negative charge on the one at the 4 position, you could get an electrostatic interaction between these two, and that would help stabilize the structure as well. That brings us to beta conformation. And beta conformation is essentially a zigzag type structure that's formed by the backbone of the polypeptide. And we essentially have 2 types of beta conformation. We have anti-parallel, and then we also have parallel. And essentially what it means is, if you look at these polypeptide chains, the first one that's shown here is where I'm sort of moving the mouse around here. You can see on the left it has an N terminus, and on the right it has a C terminus. So from N to C, you can see this arrow indicates N to C in that direction. If you look at the second polypeptide, you'll notice that it goes in the reverse direction, indicated by the arrow in the opposite direction. We have the N terminus on the right, and we have the C terminus on the left. And then if we go to the third one, it switches again. We have the N terminus on the left, the C terminus on the right. So you can see by the arrow here, it's indicating the direction's going in the opposite than the middle one. So this is what it means when it says anti-parallel. It just means that the polypeptide chains are alternating in their arrangement. But what you can see here is that the beta sheet is actually stabilized by these hydrogen bonds that occur between the hydrogen atom that's shown here and this carbonyl oxygen that's shown here. You can see there are several of them that occur between 2 polypeptide chains, and this provides overall stabilization for the beta sheet structure. That was anti-parallel. Now we have parallel beta sheets, where we have N terminus to C terminus going from left to right for all of the polypeptide chains. So in this case, they're still stabilized by hydrogen bonding, but notice that now there's a significant angle on that hydrogen bonding. At any rate, understand that you have 2 types of beta sheets, parallel and anti-parallel. Okay. Another type of secondary structure is known as beta turns. This occurs in globular proteins, and it essentially serves as a transition point between alpha helix and beta conformation regions in the protein. And essentially, you can think of the beta turn resulting or coming about as a change of direction of a protein chain. So, if you think about it in that way, it's not surprising that these occur at the surface regions of the protein. The most prevalent amino acids that are present in beta turns are glycine and proline. Glycine is small. Again, it only has a hydrogen atom as a substituent group, so it's very flexible and it can fit into small spaces when necessary. The nitrogen atom that is present in the ring structure of proline, again, it sort of restricts certain motions, and it basically forms a cis conformation, which actually helps when you have a change in direction along a peptide chain, a polypeptide chain. So, this diagram illustrates it much better than how I'm explaining it. If you look at the type 1 beta turns in proteins, you can see here that we have proline is present in type 1s. And essentially, this is the 5-membered ring that contains the nitrogen structure. This is sort of in one plane down here. And then, going in the other direction, perpendicular to that, you have the rest of the amino acids. So it sort of facilitates this bend in the structure of the polypeptide backbone in the type 1 thanks to the proline molecule. And then you can see in the type 2 we have glycine residues that are present. And again, this is just due to the flexibility of glycine allowing that. And in both cases, you'll notice that additional help for stabilizing the beta turn structure is provided by hydrogen bonding, again, between a carbonyl group and then this hydrogen that's attached here. In both cases, oxygen and hydrogen. So overall, when we talk about protein structure, the three-dimensional arrangement that we talk about, this is actually known as the tertiary structure. The quaternary structure, on the other hand, is actually the arrangement of subunits in three-dimensional space. Now remember, if a protein only has one subunit, then it is not going to have quaternary structure. If you recall, we had an example of a one subunit protein, and then we had an example of a 4 subunit protein, which is hemoglobin, for example. So hemoglobin would have quaternary structure. Another molecule such as myoglobin, which only has one subunit, would be an example of a molecule with no quaternary structure. To be honest with you, quaternary structure isn't really, it's not, I don't want to say it's not used as much, but I haven't really seen it used as much. The tertiary structure is what's really seems to be sought after the most, but the quaternary structure is important if you're talking about the relationship between subunits. Globular proteins are actually soluble in aqueous mediums, and they form a spherical or globular structure. We also talked about fibrous proteins, and these are polypeptides that are arranged into long strands or sheets. And in globular proteins, we usually find several types of secondary structure, while in fibrous proteins, they typically contain only one type of secondary structure. Now, just to get into some specific examples of fibrous proteins, alpha keratin is a good example, and this is found in hair, nails, and skin, Collagen is another example of a fibrous protein, and collagen is found in skin, bones, ligaments, and many other anatomical entities in the body. And unlike globular proteins, fibrous proteins are insoluble in water. And in part, this is because they are composed of many different hydrophobic amino acid residues, which doesn't allow them to be, dissolved in water. In general, fibrous proteins contain a structural motif, which is kind of a fancy way to say that it basically has repeating units of amino acids. So alpha keratin, which is the major protein of hair, wool, nail, claw, quill, horn, hoof, and stratum corneum. Okay, this is a pretty important molecule. It provides all of these biological materials with great strength. Really amazing. Just to give you an idea, like a hair fiber, the tensile strength of a hair fiber, if you took a piece of copper wire that was like the same diameter, it wouldn't be as strong as a hair fiber. So it's really an amazing biological structure. Types of alpha keratin, it belongs to a family of proteins known as the intermediate filaments. And, in addition to playing a biological role in differentiated tissues, and differentiated tissues are all the ones that I just described to you, consisting of hair, wool, nail, claw, quill, horn, hoof, and stratum corneum. In addition to intermediate filaments playing a role in this, they actually are present in metabolically active cells, and there they actually play an important role in the formation of the cytoskeleton of the cell, and that's spelled CYTOSKELETON. Alpha keratin is a right-handed helix. If you take two alpha keratin molecules, wrap them around each other like a piece of rope, how a piece of rope is made, or twist them around each other, you form what is known as a coil. And that's kind of what I'm showing you on this slide here. You go from one alpha helix, you take two alpha helices, and you twist them together around each other. You form what's known as a coil. And then, that coil, the alpha helix has a right-handed twist, or it's actually a right-handed helix. The coil would have a left-handed twist. If you take two coils, associate those together, you get a protofilament. Then if you take two protofilaments, associate those together, you actually end up with a protofibril. And then if you take four protofibrils associated together, you get an intermediate filament. Now in general, alpha keratin is rich in amino acids that contain hydrophobic side chains, such as alanine, valine, leucine, isoleucine, methionine, and phenylalanine. Okay? And when we talk about quaternary structure of alpha keratin, it's essentially stabilized by disulfide bonds. And further to add on about hard keratin tissues and things like that, a large proportion of the amino acids in hard keratins are cysteine or cysteine residues. Just to give you a little bit more of a better look at intermediate filament structure. So we start with the keratin alpha helix. You can see the helical structure shown here. If you take two of those, wrap them together, you get a coil-coil, which is shown here. Again, the keratin alpha helix is right-handed. The coil-coil has a left-handed twist. If you take two coils, put them together, you get a protofilament. Then if you take 4 protofilaments together, put them together, you end up with a protofibril. Another way to look at this would be, looking at the cross section of a hair fiber. Here on the exterior part of this cross section, where I'm sort of highlighting right now with the mouse, this is where you have the cuticle, which consists of a layer of a number of cuticle cells. And then inside this sort of reddish or pink color here, all of this inside is considered the cortex, and this consists of cortical cells, which are shown or indicated by the lines that are drawn in here, showing you that you have various cortical cells within the cortex. And then, what's shown is where is the alpha helix present. Notice that you start with the alpha helix, then you have the coil-coil, the protofilament, the protofibril, and then the intermediate filament. Intermediate filaments are embedded into an amorphous matrix in the structure of the hair. Keep in mind that the alpha keratin structure, or the intermediate filament in total, is located in cortical cells, so it's in the cortex. You do not find these in the cuticle cells. And I don't want you, we'll cover more in detail or with greater depth later in the course about hair structure, but I kind of just want to give you an idea of alpha keratin, how it forms the intermediate filament part, and then just to give you a general idea of where it's at. But please be very familiar with everything that we've discussed about this so far today. That brings us to collagen, which is another major structural component of connective tissue. And again, that could be bone matrix, cartilage, cornea, tendons, and even skin. And now we have the collagen sort of helical structure, which is very different from the alpha keratin helical structure. So, in the alpha keratin, or the alpha, just in general with alpha helices, the alpha keratin had a right-handed alpha helix. With collagen, we find that we have a left-handed helix. And actually, it's not really called a helix or an alpha helix. What it is called is an alpha chain with collagen, so that's different. It's not an alpha, don't confuse it with alpha helix. It's called alpha chain. And again, it sort of is a left-handed alpha chain as opposed to a right-handed alpha helix that we see with alpha keratin. And what happens with that is that this is showing you just one alpha chain here, and then this ribbon structure is giving you an idea of the arrangement along the backbone of the chain. And then in B, we have a space-filled model, or Van der Waals model, of one alpha chain. And then in C here, we actually have 3 alpha chains that are shown. And what happens with collagen is you have 3 alpha chains that are super twisted together to give you a triple helix, or it's also known as tropocollagen structure. Triple helix or tropocollagen, so it's TROPOCOLLAGEN. Okay. So going back to the alpha chain, the alpha chain, the dihedral angles are, for phi, it's negative 51 degrees. For psi, it's positive 153 degrees. In general, there are 3 amino acids per turn in the alpha chain, and the amino acid sequence follows the following scheme. You have glycine, X, Y. So that's sort of describing the repeat unit of the primary structure. So glycine, X, Y, and X is actually proline, and Y is hydroxyproline. Okay. So again, the alpha chain is left-handed. Okay, so that's different than alpha keratin, which is a right-handed helix. And then when you have the 3 alpha chains super twisted together, it's a right-handed arrangement. Okay, and that's unlike alpha keratin, which is left-handed when you have the coil-coil structure. And then if we look at the fibrils a little bit differently, we can look at them with an SCM micrograph. So here we're looking at, the collagen fibrils here. Okay, and what you can see are these bands that are characteristic. These bands that you can see with the SCM or the scanning electron microscope image. And these bands are essentially due to the alignment of the alpha chains. And to give you an idea of that, what do we mean by that? So this is showing you various chains that are various collagen molecules. So if you look, like you have 3 alpha chains that are super twisted together here, and then so each one of these things that you see in this diagram where I'm pointing the mouse right now, this is actually a triple helix structure. So we have a row here of triple helix structure, and then that's connected via crosslinks, which are shown here, to the next row, and then those are cross-linked to the next row. So you're forming like this hierarchical structure of collagen structure. But what you'll notice is that the peptide chains don't always end and complete, or end and finish and then begin with a new chain in the same place when you go from like the bottom row to the next row to the next row to the next row. But where you see the striations at, those are actually caused by what you can see here in this pattern that's shown here. So it's caused by this gap that occurs in the structure. So, collagens, as I mentioned, the triple helices are actually, their strength, or they're reinforced by cross-linking that occurs between chains. There are several types of cross-links that occur in collagen, and usually they involve lysine residues. Lysine can be converted to alysine, which is spelled ALLYSINE, and then that actually can form the crosslinks. And here we show 2 other molecules that are important in collagen crosslink structure: pyridinoline and glycolaldehyde pyridine. These are also important structures that basically provide cross-linking points between the triple helices. In addition to collagen, the skin dermal matrix also contains another very important protein that is known as elastin. And elastin, as you can guess from its name, it implies that it's a polymer or a protein with physical characteristics of a rubbery material that snaps back to its original form when stretched or compressed. So in general, elastin is composed of mostly glycine, alanine, valine, and proline. And the three-dimensional structure of elastin actually lacks periodicity that we see in other fibrous molecules. So it's a little bit different. And, to complicate things more for elastin, it doesn't have an organized secondary structure. But crosslinks are present in elastin, and these are formed by lysine aldol. So again, that's spelled Allysine space aldol, Aldol. They're also formed by desmosine, which is Desmosine, and then isodesmosine, and then also lysinonorleucine, which is spelled, it's all one word, Lysinonorleucine. So now, before we continue on, there's one little section that I'd like to discuss vitamin C deficiency. And just to give you a little bit of history on this, vitamin C deficiency was very common in the past. If you think back to the period during the European discoverers and the conquistadors, just roughly between the years of 1400 to 1800 AD, this was a period when fleets of ships sailed around the world in search of treasures, wealth, and new undiscovered lands, at least to the people that were trying to discover them. And, during these expeditions, the sailors were at sea for extended periods of time, and they would often become ill with a disease that's known as scurvy. It's spelled Scurvy. And this disease leads to spots on the skin, tooth loss, damage to gum tissue, and also impaired mucous membrane function. And it wasn't actually until the middle of the 18th century that the Scottish surgeon James Lind, who was in the British Royal Navy, he discovered that there was a connection between scurvy and the consumption of fresh fruits and vegetables. And essentially, sailors who did not obtain vitamin C in their diet were not able to synthesize collagen. And, nowadays we understand the molecular mechanisms behind this. And essentially, there's, one thing that needs to take place during collagen synthesis is you need the hydroxylation of proline to form hydroxyproline, and this requires an enzyme known as prolyl 4-hydroxylase, and that's spelled PROLYL space 4-hydroxylase HYDROXYLASE. And this enzyme, prolyl 4-hydroxylase, actually needs vitamin C to fulfill its duties, which again is to hydroxylate proline, forming hydroxyproline. And there are some more details associated with this process, but I think we'll kind of keep it to that for our discussion in this class right now. So the next thing I want to talk about are globular proteins. And globular proteins, again, we talked about fibrous and globular proteins, and we know that they are much different in terms of their structure and function. Globular proteins are spherical in shape, and again, they play various roles such as enzymes, They regulate cell processes. They serve as antibodies of the immune system, and they also are important transport molecules. For example, hemoglobin and insulin. So, as an example of a protein's three-dimensional arrangement, we have a globular protein that's shown on this slide, and this is actually interleukin 12. Interleukin 12. I don't think that's spelled out here. Interleukin is spelled INTERLEUKIN-12, and you can see it's indicated by IL-12 here. And again, this is just to show you protein three-dimensional structure, but we're using a protein that's pretty important for skin as an example. So interleukin 12 actually belongs to the cytokine class of molecules. Cytokine, you can see spelled on this slide, is given at the top of the screen. This is a family of molecules that are proteins and peptides, and they're essentially cell signaling molecules that are secreted by particular cells in the body. They travel through intercellular space, and then they bind to the surface receptor of a target cell. So, we mention it with respect to the skin because skin cells, which are keratinocytes, skin cells referring to the epidermis, has keratinocytes, and keratinocytes actually are very well known for secreting cytokines into extracellular space, especially during times when there's a need for an immune system launch. The keratinocytes play an extremely important role in that. They secrete cytokines very often, and that helps regulate a lot of the more macro processes that occur in skin. Even when you have wound healing, or even if you just have foreign invasion by a pathogen, those keratinocytes are launching their defense by secreting these cytokines, which go to other cells. It can even go to cells in the dermis or immune cells, and they sort of tell it, we need to do this, and either to help launch this attack or defensive or whatever it may be. Okay, this particular cytokine, interleukin 12, is actually expressed at high levels in people with psoriasis, and psoriasis is spelled P-S-O-R-I-A-S-I-S. It's an immune-related disease which results in the overproliferation of skin. It ultimately leads to scaly patches that are distributed over the skin surface. So again, the three-dimensional structure of interleukin 12 is shown on this slide, and it nicely illustrates three types of secondary structure in protein. So we're looking at the overall three-dimensional structure of the protein, but you can also see the secondary structure of the alpha helices that are present here on the right, given by this sort of helical type structure. We can also see there are beta sheets present here, and again, interestingly, you have beta sheets. The way this is illustrated, you have beta sheets going in one direction, then you have another polypeptide chain in the opposite direction, and then another one in the opposite direction. So, this series of beta sheet here would be in the anti-parallel form. Over here you have some other beta sheet segments within the chain. And then we also have beta turns, which if you look at the area where I'm tracing now, that's given by this sort of, I think it's a green color, and it's a really thin line. That's basically giving you, showing you where the backbone of the peptide chain follows this beta turn type structure. And then, just to finish off this section, I'd like to talk about the determination of protein structure. And, you know, protein structure is typically determined by two methods. One of them is X-ray diffraction. The other is nuclear magnetic resonance, also known as NMR. And, you know, both one-dimensional and two-dimensional NMR are used to elucidate structures of proteins. But nowadays, you can access data on the three-dimensional structure of proteins by just going to the protein data bank website. And, you know, there's numerous data giving the three-dimensional coordinates of all the atoms in proteins for many different proteins. And essentially what you're looking at here is I obtained the data for interleukin 12 on that website, and then I actually graphed it, or, you know, plotted it or generated this three-dimensional model using a software package. This particular one was called MacPymol, which is by Delano Scientific, and it nicely, you know, puts it into this very nice arrangement for you and organizes it, you know, and you can see where the alpha helical sections are, where the beta sheet sections are, and where the beta turn sections are. Okay, so that finishes up chapter 2. If you have any questions, please do not hesitate to contact me, preferably by email, and I'll get back to you as soon as possible. And, you know, I realized that we really, you know, chapter 1 was probably a little bit lighter in terms of what type of material we covered. Chapter 2, we're really starting to dig into some biochemistry at this point. Again, you know, try to please keep up with the material, because we're going to be covering quite a bit of material, and you really need to cover it frequently in order to keep up and be able to, you know, be successful in this class. So, you know, please reach out to me if there's any questions, or if there are any questions or concerns. Okay, thank you.